So today we're going to begin our study on the cell structure by discussing the cell membrane. The cell membrane is really instrumental to actually making up our cell. Without the cell membrane, our cell wouldn't exist. And so we really today are going to focus on what makes up the cell membrane and what are the different components. But you do want to keep in mind some of these essential questions that eventually we'll want to be able to answer all of. When we're looking at the cell membrane, we do need to know some different names that are used to describe it, okay? Very basic, we all have heard of the cell membrane. And another one that a lot of us have probably heard of is the plasma membrane. And that's just referring to the fact that it's actually holding in that cytoplasm, that plasma, within the cell. Remember that within the cell, you have water, aquatic, okay? It's liquid. And outside the cell, it's also aquatic, also liquid. And that's going to be important to keep in mind after when we talk about how the cell membrane actually forms. So that's the first name. The second name that you'll hear a lot of is the phospholipid bilayer. So 2 layers of those phospholipids are actually used to form the cell membrane. And what we need to know about that is that actually forms spontaneously. And we'll discuss how that happens in just a little bit. But the third name is called the fluid mosaic model. Okay? And we're really going to concentrate on these 2 names today. The phospholipid bilayer, how does it form? Why does it form that way? Or why is it structured this way? And then the fluid mosaic model, fluid referring to the fact that it's not stationary. All these molecules within the cell membrane are constantly in motion, however much or however little that motion is. And mosaic referring to all those different components, just like an art piece, an art mosaic, lots of different ceramic tiles creating a big picture. But no matter what you call it, one of the biggest and most important things to remember about the cell membrane is the fact that it is permeable. Okay. So different or particular molecules are able to move in and out. Some of them can move in and out without help, and some of them will require some help of an outside source. Within the cell membrane, the pieces that we're going to talk about, the different parts of that mosaic that we're going to talk about, will be the actual big proteins themselves, the individual phospholipids, which you should be familiar with the shape of by now. We'll talk about the cholesterol, that yellow piece within the cell membrane we've also mentioned before. And then we'll also be talking very briefly about these carbohydrates that actually act as IDs or identifiers of the cell. Okay? So right now we're going to go ahead and talk about those different components of the cell membrane. The main parts of the cell membrane, first of all, is going to be your phospholipid. Okay? So your phospholipid, remember, is composed of 2 parts. You have your phosphate head, and your 2 fatty acid tails. Remember, the phosphate head was one of our functional groups. It was that PO4, and it had a negative charge to it, and it was a polar molecule. Okay? Whereas on the other hand, our fatty acid tails were a chain of hydrocarbons. Okay, carbons either saturated or unsaturated with hydrogen, and these were non-polar. Okay, because we have this polar and non-polar region, remember that this molecule is amphipathic, it's both. This is actually what allows it to spontaneously form our cell membrane. If you took a bunch of phospholipids and you threw it into some water, they can actually congregate within each other and actually have all of the heads face the outside with the fatty acid tails on the inside. Okay? When we're actually forming the cell membrane, the same thing happens, except we're forming a bilayer, 2 layers. We have the heads on the outside touching the cytoplasm, the watery fluid. We have the heads on the outside touching the water outside of the cell. And those hydrophobic tails are going to be on the inside, hiding within the membrane. So hydrophilic heads outside, hydrophobic tails inside. And that's how we form our cell membrane. Now, you do want to keep in mind that those saturated and unsaturated tails do affect the fluidity of our cell membrane, our fluid mosaic model. So here we have our unsaturated fatty acid tail. And remember, that's created through the carbon-to-carbon double bond usually. And then we have our saturated tail, which has no kinks in it, no double bonds, and it's actually going to make the membrane a little more rigid. Okay? So the unsaturated, the kinks create the fluidity, the saturated keeps it more rigid. One thing to keeping that in mind, the fluidity of the cell membrane can be regulated through those fatty acid tails. Just like with a car in the wintertime, you're going to want an oil for the car that's more fluid, less viscous, okay? Because the temperature is slowing it down, so you want something a little more watery so it can move a little more easily. And whereas in the summertime, you might use an oil that's more viscous, thicker, because the heat's already going to create those molecules or cause those molecules to move more. So same idea with our phospholipids. The next structure that we want to take a look at is going to be our cholesterol. So number 2 is our cholesterol. Cholesterol is also going to increase the fluidity within our cell membrane. And it's actually acting as like a little bumper within those phospholipids, creating a little more space between them. And that's going to increase the fluidity. Thinking a little bit about the chemistry of our cholesterol molecule, we have our hydroxyl group here, which is polar, and our nonpolar hydrocarbon chain. And this is really similar to that polar and nonpolar phospholipid as well. And so it's actually going to have the same type of behavior. Some of the molecule, or the hydroxyl OH group, is actually going to be sticking out on the outside by the phosphate heads. Whereas your hydrocarbon chain is going to be nonpolar, so it's going to want to stay inside the hydrophobic. And all of this is helping to increase the fluidity. Then the third portion is going to be, or third component we really want to talk about, are the proteins. There's 2 types of proteins that we'll talk about. Integral proteins that are going to stay within the cell membrane, and then peripheral proteins that are going to stay on the outside. So out, in. Okay? And so on the next page, we're going to actually look at those specific proteins, or actually some functions of those proteins. So proteins have many, many jobs. And within the protein, we have, well, when we're looking at those proteins, we also want to think about how we have our external and internal portions of the cells. So just like our cholesterol and our phospholipids, the chemistry of the protein is going to determine what part of it can be on the outside, what part of it has to stay on the inside, what's polar, what's non-polar. Okay, so you can't forget about that. And then right here we'll have our selectively permeable membrane. So thinking about the functions of our proteins, the first function is to act as a transport protein. Okay, transport protein. These are going to be specific in shape and specific in charge. So again, the chemistry of the molecule is involved. The second type of protein you have within the cell membrane are enzymes. There are enzymes that are on the outside or inside of the cell membrane that can catalyze different reactions. And again, all of these are going to be specific if you notice the different colors that are being used. And the third function is going to be to act as a receptor. Again, these receptors are going to be shape specific. They can receive a molecule and cause a reaction. So here's our signaling molecule, and here's the action result of that molecule. Our fourth type are going to be proteins that can be used as IDs. Here you have a protein with that carbohydrate chain attached to it, and we're actually going to call that a glycoprotein. Glycoproteins actually allow the cell to recognize itself, self-to-self cells, which is important when we think about the immune system. And then we also have those carbohydrate chains that are attached to our lipids, and we call those glycolipids. And glycolipids, we'll talk about more later on, are actually going to recognize tissues, so recognizing blood type within the red blood cells. The fifth type is actually going to be adhesion proteins. Okay? These proteins are actually going to provide stability to our cell membrane. They can actually adhere to neighboring components, or they can actually adhere to other cells to create more stability. And then the last type is actually going to be our surface proteins. Our surface proteins can actually act as anchors. And here they're actually attaching providing an anchor to the cytoskeleton of our cell. Okay? So here are the functions of our proteins, 6 main types. You want to make sure you look at those review questions that are underneath. But keep in mind that the cell membrane has all these different components to it that are allowing it to function and allowing the cell to help maintain homeostasis. So make sure we review over these proteins, the phospholipids, and also looking at those cholesterol molecules.